We're here with Professor Margaret DeAngeles from the Moran Eye Center at the University of Utah. Welcome, Professor.
Welcome.
We last spoke to you at the Harvard AMD conference in October of 2014. Tell us a little bit about some of the exciting work that's been going on since that conference.
Okay. Since that conference, we had to validate our work and do some confirmatory studies. So we're in the process right now where we're getting ready to publish our work. And we've confirmed known pathways in age-related macular degeneration, particularly for the intermediate form, the form that happens before we go on to the late stages. And that's the form we want to go after, because we want to arrest progression and try to intervene in that form of the disease. But we've identified new genes in those pathways that could potentially be therapeutic targets. And for that, we're excited. And because we're showing function, not just gene associations. And the idea is that the activity of these genes is different between those with disease and those without disease. And the thing that's nice is we have a team of collaborators with different areas of expertise, retina specialists, people with backgrounds in statistics, in molecular biology, and biochemistry that can work together and have different perspectives in bringing that together. So that's exciting. We hope it'll lead to therapeutics for intervention.
There are many, many genes that are implicated in AMD. What specific genes have you identified that you're really enthusiastic about?
I can't say what those genes are, so you have to cut that out. Okay, so genes that are involved in lipid metabolism, cholesterol biosynthesis, and genes that are involved in neuronal pathways that have some overlapping pathophysiology with age-related macular degeneration.
Do any of these genes look like they might be single gene targets for gene therapy?
I wouldn't do single gene targets. I would, because of the redundancy of our genome. And what I mean by the redundancy of our genome is you have genes that have more than one function. Okay, and genes that function in more than one pathway. So it would be smart to go after 3 or 4 genes at a time. And that's not a new idea. That's been done for different forms of cancer, for different forms of other diseases, complex diseases like age-related macular degeneration, to halt progression of disease.
Tell us about some of the other work that your lab is conducting.
And that's just to do with levels of gene expression. Other work that our lab is conducting is looking at populations with different ethnicities and seeing why certain populations don't get any age-related macular degeneration. And we have papers under review. They're not yet accepted. They're under peer review. And to see why they don't get any disease at all. Some of that we think is due to their diet, but more importantly, they have a high level or frequency of protective alleles in some of these genes. And that's quite interesting when you compare their ethnic background to other members that live in different parts of the world, say people from Utah or Boston, or people that live in central Greece or South Korea.
What are the names of these protective alleles?
They're found in the complement factor H, which is well known to have both protective alleles and alleles that increase risk for the disease.
What is the genealogy of these people that have protective alleles?
The genealogy of these people is that they're from the oceanic region, which is an island that's found off the coast of Australia that's near Indonesia, so an isolated island. And we try to figure out what their ethnic background and where they may have migrated from.
Well, that's fascinating. This is a population that was essentially cut off from the rest of the world's population for maybe hundreds, if not thousands of years. Or are they linked to the mainland? Is this a traditionally a completely island population?
Well, the thing that was interesting about this population, this is they're the Timorese, is that while they are predominantly oceanic in nature, there is European ancestry mixed in, a little Han Chinese, and they do have varying backgrounds. But also they keep a low body mass index, so that might have something to do with it too. So it may be a mixture of the environment and also their genetics. And we'll have to explore that more. But right now, we think it's maybe their genetic background, but also their ethnic background, but more importantly, the high level of protective alleles.
In October, you spoke about your work developing an eye bank, collecting eyes. Tell us about some of the applications of that eye bank.
One of the applications of that eye bank is the work to study the gene activity, which is the first step in studying function of genes. It's one thing to get gene associations and say this gene is associated with risk of disease, but that doesn't tell us anything about disease causality. And for us to really develop a therapeutic target, we need to understand and to get to causality, we need to understand mechanism. And understanding activity is a first step. And the gene bank, the eye bank helps us to do that because it helps us to access tissue where the disease is actually taking place. And the tissue we are able to get due to our wonderful donors, our eye donors in the Salt Lake City area of Utah, we couldn't do that without these wonderful people, and the Lions Eye Bank, is because we're able to get fresh tissue. And that means we're able to get tissue that is accessible in under 6 hours post-mortem. And that's very important because tissue begins to degrade after 6 hours. And it's not very useful to do protein studies and studies of gene expression. So it's ideal for that. And you can access what we need for age-related macular degeneration, that being the retina and the retinal pigment epithelium in the choroid. But more importantly, we're also able to get blood and serums from the patients so we can make correlations between circulating markers, biomarkers, and what's going on in the eye. So it creates a very nice repository of biospecimens for study. So we can take steps beyond just studying DNA and genetic association.
So if there are family members or individuals that are considering a donation, what is the process that they need to undergo in order to let you know about it?
They can contact me directly, and I can provide that information, or they can contact the Lions Eye Bank. But I'm happy for them to contact me directly, and I can facilitate that.
Turning back to the functional aspects we were talking about earlier, what are, in your mind, the closest or the most predominant mechanisms that you think are most responsible for the disease causation?
Causation. Well, I think it depends on the subtype you're studying. My lab hasn't even begun to study the atrophic form, the most end-stage dry form, but a lot of labs have studied that. I think there's a lot of work being done in the immune response. A lot of groups are studying that. I'm sure that has an important place. I think groups are studying angiogenesis. My lab in particular, as our other labs, are studying the whole cholesterol hypothesis, lipid metabolism. There's groups studying overlap with neuronal pathways involved in Alzheimer's disease. Our group is certainly involved in that with my collaborator, Lindsay Farrer. And so I think those pathways are major pathways that groups are going after. And certainly there are other pathways which I haven't touched upon that I'm sure are very important that other groups are studying.
Turning to translational aspects, our patients always ask us, when are we going to see some results of the work that's being done, and what are the implications for them of this work? Could you share your perspective on that?
My perspective is that I hope to solve this disease in my lifetime. And I want to stress again, this is a disease that runs in my family. So I'd like to have some form of therapeutic intervention in my lifetime, not only to help my family, but to help those around me. Because remember, if you have a first-degree relative that is a parent or a sibling with this disease, you're at 6 to 12 times more likely to get the disease than somebody from the general population. So my perspective is that we will solve it in my lifetime. So I like to take a conservative approach. And, get regular eye exams, particularly if you're over the age of 50 and over, and if you have somebody in your family with the disease.
That's very powerful and promising news. Would you like to add anything else, Professor, about work you're doing and other prospects going forward?
Not right now, but just have a really good team. No man is an island. Have a really good team, and I work with a really good team of people, and I have a wonderful chairman, Randy Olson, at the Moran Eye Center, and a good group of people I work with. And thank you. And thank you to all our patients and donors that help us.
Thank you, Professor.
Thank you.